Good afternoon, everybody. Thank you very much for the introduction. Well, we listened a lot about pharmaceutics, drug formulation, and also drug delivery systems. But I'm going to talk about something which is on the other side of the coin, to be able to, if we know a drug which actually works in the in vitro system, how can we test these in vivo models? I specialize in arthritis. My field is rheumatology. I actually did my pharmacy before, but then I actually moved into medicine. And I work on osteoarthritis, which is the most common form of arthritis. Now, osteoarthritis is out of all of you, including me, at one stage of our lives, we will all get osteoarthritis. So it's not a good disease to actually have, but it's the most debilitating disease, and we have to counteract it in one way or the other. So what we have done is we've devised some tools to be able to understand what the drugs does when it actually is injected into a joint, if you have arthritis, where it travels, and what it actually does in terms of its mechanisms of action. So the two possible therapeutic options which we are right now looking at is the stem cell therapy, where we are actually injecting stem cells right into the intra-articular space, which is right either in your knee as well as in your hip. Or we are targeting this particular concept, which is the autophagy pathway. The autophagy pathway is the pathway which is the autophagy pathway is a pathway where you can, when you promote this pathway, you're able to increase the survival of the cells within your tissue, whether you are doing through gene therapy, whether you're doing through stimulating with certain pharmacological agents, you're able to actually stimulate the autophagy process. So these two therapies we are currently looking at in terms of treating patients who have osteoarthritis. Now, as I said, you have 68% of women which are actually affected by osteoarthritis, and lower in men, fortunately for us, but for women, it's much higher incidence. Now, if you look at to be able to deliver a drug inside a joint, it's really, really important to be able to understand the actual joint structures which are affected in osteoarthritis. If I open up my knee, it's going to actually look like this, where I have a cartilage which looks like normal here, and if you see inside this cartilage, you have lots of these cells. And it's really important because when you actually inject a drug within the joint space, the drug has to literally interact with these particular cells to be able to promote the health as well as maintain these cells for a longer period of time. And you know, we all age as we grow older, but in our body, the cartilage is in such a way that it tends to age a lot faster than our body. That's why it's very, very important to be able to maintain this with certain pharmacological agents as well as certain natural therapies, as previous speaker said. Now, during osteoarthritis, what happens is you get this destruction of the cartilage, your bone remodels, as well as you get a lot of inflammation into the synovium. And that's the problem. When you inject a drug, because you have so much of inflammation into the joint space area, that's why a majority of the therapies do not actually work. Because by the time they are injected, they don't have, for example, if we are doing a gene therapy, and we're trying to alter the homeostasis of the synovium, which is a joint structure area, it's impossible sometimes to be able to deliver the drug at the targeted site. Now, from past few years, we've been trying to understand these complex networks, that if we are going to deliver drug, and what's going to happen and what it's going to actually interact with. Now, if I actually draw a systemic diagram of a joint, and I look at, this is bone, and this is cartilage, and this is what I call synovium. You have all these different cell types, inflammatory cells, macrophages, neutrophils, and fibroblasts, which are all encompassed into the joint area. And as a result, you have this mesh network and very complicated system where when you actually inject a drug in this joint space area, it has to counteract with all this plethora of cytokines and all these destructive mediators. Now, so to be able to understand the delivery of the drugs as well as to be able to understand the mechanisms of disease, we've been working on animal models of osteoarthritis. And why we do animal models of osteoarthritis is because we have three-stage studies which we perform. We use small animal models, we use large animal models, and then we test the drugs in humans. Now, today, because it's for the context of today's conference, I think it's more relevant to just talk about the small animal models of osteoarthritis. There are different models which you can use. There are certain animals which can spontaneously get osteoarthritis, but you can also induce arthritis in certain animals. And these are really important models, because when you institute or induce a model, you can direct a particular therapy whatever way you want to, whether you're giving it into the joint or whether you're taking it orally. You can track what's happening in the area of interest where you have actually induced this model. And then, of course, you have genetic models, which we have created. My lab has created almost around 7 genetically modified mice. When you delete a gene of interest, these mice actually get osteoarthritis. And then by targeted delivery of these genes into the joints, you can actually cure osteoarthritis. But that is a trick, because it works in animals, but it actually does not end up working in humans. And we've tried 2 gene therapies approaches, and none have worked right now in clinical trials with patients. There is a gene called PPR gamma. I'm sure everybody whosoever has diabetes would probably know this gene. This gene is involved in insulin sensitization in our body. If I take this gene out of my cartilage, my cartilage is not going to look like this, it's going to look like this. However, if I actually reintroduce this gene back into the cartilage, my cartilage will revert back to a normal cartilage. And this is only, like I said, can only happen in animals, cannot happen in humans, because the delivery system and also the complicated diffusion process within the joint is absolutely complicated in a human being, whether it's a large size of the joint as well as the diffusion ratios. Now, the model which we developed in our lab, and I was involved in creating this model, was a model called destabilization model of osteoarthritis. Now, this model is being used worldwide to be able to test drugs as well as to do an intra-articular therapies as well as stem cell therapies, where basically you're opening up the joint and you are basically taking it's called medial meniscus in your joint, where you're actually transecting the medial meniscus, and as a result, the joint becomes destabilizes. And as a result, you're walking, you have mechanical tension, and because of the sudden rubbing of your top part of your joint, so basically it's called tibia or femur, when you actually rub them together, it suddenly creates osteoarthritis over a period of time. And it's a good model because osteoarthritis develops slowly in these mice, and as a result, you can tailor when you want to inject the drug. Do you want to inject the drug when the osteoarthritis has not taken place, or when the osteoarthritis has already established in these animals, and then you inject the drug and see what the mechanism of action is, and also if these targeted therapies actually work? Like I said, this model is really good because you can tailor when you want to do the delivery. You can also see the kinetics of, this is a cartilage. The lower part of the cartilage, upper part of cartilage, this is like no surgery. But when you do the surgeries, you can see, you can actually see the destruction of the cartilage. So basically, when you introduce a drug, you can really see where it actually would go, and if it's able to actually reconstruct this particular area of the cartilage. I'm just going to skip this because this wouldn't be that relevant. So what we are doing right now is to be able to use these models and to be able to understand how cell therapy and how cell therapy could be possible in escaping or limiting or reducing incidence of osteoarthritis. What we've done is, due to intellectual property as well as patent, I can't actually tell you exactly what we are mixing and what we are actually administering, but I'll just tell you. So basically our body is made up of cells, and these cells are coming from certain source, and one of the sources are the stem cells. And these stem cells have the ability to regenerate. Now, what we're doing is we're taking these stem cells, and these are coming from umbilical cord. So basically, whenever you have a birth of a child, the umbilical cord, which is disconnected, is taken, and that umbilical cord is processed to be able to isolate these stem cells. Or you can do that from your bone marrow, or you can even take any tissue and you could actually derive these stem cells. Basically, you take these stem cells and you are putting this with hyaluronic acid. Hyaluronic acid has a lot of properties in itself. Patients who have osteoarthritis or rheumatoid arthritis, normally you inject hyaluronic acid in that. So basically what you're doing is you're mixing stem cells with hyaluronic acid and you are injecting it into the joint space. And what we're doing is we're doing two comparative studies. We're doing one in a mouse, and we have the first clinical trial now ongoing, the first in North America, where we are testing the efficacy and safety profile of these stem cells injected in patients who have osteoarthritis. And what we're doing is we're using a tracking system by labeling these cells so they have color. And when you actually inject them, you can do in vivo tracking of these cells. You can see where these cells are going into the joint space, what they are doing over a certain period of time. Of course, if I'm injecting the cells today, after 3 days or 4 days, I'm not going to be able to see any of these cells into the joint space. However, at a certain time range, for example, if I'm injecting now, for first 24 hours to 48 hours, you're able to track these cells. So it's a good system to be able to track the distribution of these cells. So like I said, what we're doing is we're taking these cells, putting them with hyaluronic acid, and right now we're looking at the effects of this and understand what the distribution of these drugs are. We also are working on an adenoviral system where it's an agent, it's a gene called ULK1. ULK1 is a gene which is, if you don't have this gene, your cells die very fast. If you want to increase the survival of the cells, you can inject ULK1, it promotes survival of cells. That means in my cartilage, where cells normally tend to die very fast, if I was going to inject ULK1 into my cartilage, is going to promote the cell survival. And that's what we are testing right now in, not in humans, but we only are doing animal testing right now. And in the first part of the results were unsuccessful, and we don't know at the moment if we are going to be able to successfully able to deliver ULK1 into the joint. And the problem is the adenovirus which we are using may not be the good adenovirus to be able to penetrate into the cartilage to be able to do the biodistribution of this gene of interest. Therefore, it's really important for us to be able to figure out how we're going to inject it, what we're going to mix with, and what the formulation and what route of administration would be. That's it. So this is what we're doing right now, and I hope to have certain results. Hopefully the clinical trials from the patients which we have is going to actually be out hopefully in 6 months. We have 2 patients right now which were recruited. So basically what we're doing is we're taking cells out of patients, and we are culturing them in incubators, and basically taking those cells and delivering it back into the joint of these patients with hyaluronic acid. And we don't know at the moment if this is going to be efficacious, but of course our animal models do suggest that that it promotes, it decreases the inflammation of the joints. However, we don't know how it's actually going to work in human beings. So this is my research team, quite a young people who have worked immensely hard to be able to come up with all of the work which I just showed you. I just showed 1% of the work we do. We do a lot of gene therapy, we do a lot of animal models, we do a lot of clinical trials, and this is all my collaborators and funding agencies who have helped me establish this research group. So thank you very much.
Question, please. Yes.
Thank you very much for this nice talk, fellow Canadian.
Absolutely.
My question is, I'll ask you from my expertise with the tissue engineering of cartilage repair. So maybe it's going to be naive because you're the surgeon, you're the expert there. What are you trying to fix here? Is it the loss of cells to fix things with osteoarthritis? Because I can comment on this after hearing your answer.
So the problem with so I'll talk about humans. In humans, cartilage cannot be regenerated. So there is no regeneration of the cartilage. It cannot take place. You come to a certain level where you cannot go back. Now, what you can do is you can stop inflammation, which can further reduce the destruction and the pain associated with osteoarthritis. So what these cells are doing is we are modulating these cells to be able to do not the regeneration of the cartilage, but to be able to reduce. For example, you go into surgery today and you need to have some kind of pain medication. Instead of giving any kind of pain medication, non-steroidals or celecoxib, anything, we rather actually have an injection of the stem cell, which is coming from your cells and going into your joint, which is not going to regenerate your cartilage, but it's actually going to reduce the inflammation which is associated with your surgery. Other way around, if you or I, we're all growing old and our joints are all going to actually have arthritis sometime or the other. If we are going to be able to promote the survival of these cells which tend to die faster with some means, by intra-articular delivery of certain agent, we don't know yet. In animals it works. We are able to increase the survival of the cells. But in human beings it's very complicated. The cartilage is thicker. It's very hard to do a delivery of anything into the cartilage. So it's very tricky. So right now we're only looking at modulating inflammation and pain associated with arthritis.
Actually, our answer makes me happy because otherwise, if we are doing tissue engineering for the cartilage and the repair itself, we shouldn't really be doing it. So, but the question here is, so what would really, because in the cartilage repair where there is no regeneration for the cells that are really lost, you are using stem cells that in the hope that they will really regenerate the dead cells. In your case, you are targeting the inflammation in situ, which is in the local microenvironment of the knee. So what would really the stem cell mechanism do with that?
Yeah. So I wish I had a very concrete answer to this. I actually don't have a very good answer to this question, and this question comes up all the time. You inject the cells into the joint, and it doesn't even go to the cartilage. A joint is like this, and you have your cartilage, and on the side you have your synovium. Synovium is where, if you have rheumatoid, your synovium becomes really inflamed. Now, if I actually inject a cell or anything, a drug, it all diffuses into the synovium. But within the synovium, these cells do become resident cells. And that's where they act as anti-inflammatory released agents.
I'm sorry to just be out of curiosity. Have you really checked the level of the cytokines there? Because I know that
yeah. Absolutely.
major player there.
Absolutely. We have done a lot of, we use multiplex systems to be able to do this. We have done 50-plex assays where you're actually doing 50 cytokines at the same time in single samples. We have done it in tissues, which are taken from patients as well as from mouse, as well as from dogs who have surgery as well. And we've also done in the synovial fluid from patients as well as from these animals. What we're doing really right now is to be able to, when the patient comes into the clinic, we take the blood, we'll take the urine, we take the synovial fluid, but we also call them every 3 months. Any therapy we are doing, we are tracking them longitudinally for a certain period of time to be able to see how the, whatever drug they're taking, whether they're taking non-steroidals, whether they're taking steroids, if that is working or not.
In chronic, yes, I have a question. Is chronic osteoarthritis, the synovial fluids are damaged, and that's why there is a pain. And now the only two treatment is hyaluronic acid injection, hyaluronic injection, and this knee replacement. This hyaluronic acid, as you said, the stem cells are combined to this hyaluronic acid. Is it develop this synovial fluid, or how long it will be effective? Because this hyaluronic acid injection, it's mostly effective 1 year, 2 years. After that, ultimately the patient has to go for this knee replacement.
Yeah.
So how long it will be effective? And whether this knee replacement in future, as per your planning and research, this knee replacement can be avoided by your cartilage formation or somewhat.
Again, I wish I had the answer. Really, I think joint replacement is patient by patient. It depends. I can have a very healthy joint, and somebody who's just same age as me can have a very unhealthy joint, and we both may not end up getting surgery. Or one person gets surgery, other person doesn't need to get, doesn't reach that stage to get the surgery. So it's very hard to tell the kinetics of destruction in our body. My body, that's why most of the time we are now using personalized medicine approach to be able to come up with this plan. We are using not only to be able to use different delivery systems, but also to be able to use informatics to be able to understand if I'm going to respond to a particular therapy or not, if this drug is actually going to work in me or not, and what is the actually incidence of success rate of a particular drug in a particular human being.
My specific question is the durability. Suppose if you have hyaluronic acid along with this stem cell,
yes.
if you give it, how long it will be active? And what will be the cost factor, of course, the same?
Oh, sorry, no idea. No idea. Absolutely no idea, but yes.
Also human variations.
Absolutely, human variations. Good?
Okay, it's a research-based work and a very recent, yes, very excellent work. I hope this it will open up the new vista in this stem cell engineering of this, particularly this area, and a lot of females, particularly suffering from this osteoarthritis. Most of those who are the obese patients can join these. They will be benefited from your research output. Thank you very much. Appreciation.